Greeting of the students. So, in today's lecture, we will start with the previous point, that is the growth. Now, there are different aspects we can learn about the growth of bacteria. Now, in last lecture, you have already studied the pure cultures. Now, I repeat the same thing, that is, what do you mean by pure culture? Now, when we have only one type of species or strain with us, we are called that culture as a pure culture, or only one type of bacterial species with us, then it is called as a pure culture. Now, most patient specimen and the environmental sample are containing different types of bacteria. At that time, that culture is called as the mixed culture. Now, how do we get the pure culture? There is one method to get the pure culture, is the strict plate method, by means of which you can separate out the mixed culture of bacteria and convert it into the pure culture. Now, there is a colony formation. Now, what exactly the colony formation? When bacteria can grow themselves in a multiple amount, then they are generally used to form colony. They are bind with each other. So, the population of cell arising from a single cell or spore or form or group of attached cell are called as colony forming units. Okay. So, approximately 1% of all bacteria can be successfully cultured. So, this is called as the pure culture. Now, the next point is how to prepare this pure culture. Now, this can be the practical part. We can see it in a practical, but for your very betterment, you can see that there is a burner. In that burner, we are sterilizing the wire loop. Then after cooling that wire loop, we have to put that wire loop into the inoculum where we can have the bacteria. Then we can stick that loop on the solid media in a petri plate, and then after again sterilize that loop, again go for the second quadrant, or we can say that spread the bacterial culture into the further space, then again sterilizing the loop, and final streaks are made on the media. After incubation of 24 hours, you can get the picture of 8 number, that is isolated colonies are developed after incubation, where you can observe that in first two quadrant there are dense material or dense bacterial population is there, and in third and fourth quadrant you can observe that there is a single colonies are there. Now, these single colonies can form by means of a single type of bacteria, and it is the simplest method to pick a single colony and form a pure culture of that bacteria. So, after that, how to preserve this bacterial culture? Now, it is quite difficult to find out a pure culture of bacteria. Then, it should be the preservation. That is the most important part, where we can preserve the bacteria for a longer period of time and for further use. Now, in case of deep freezing, the rapid cooling of pure culture in a suspension liquid to -50 to -95 degrees Celsius, good for several years. And the second one is the lyophilization. We are also called it as freeze drying, where the lyophilized culture is frozen at -54 to -72 degrees Celsius and dehydrated in a vacuum. So, it is also preserved for longer period of time, like years. Now, let's start with the growth of bacterial culture. Now, in previous sections, we have already discussed about there are different factors which are generally affect on the growth of bacteria. Now, what exactly the growth of bacteria? What exactly the stages of growth of bacteria, and what are the bacteria can do by their lifetime? A very first thing is, if we can call about the bacterial multiplication, the very first thing come in a mind, that is the binary fission. So, your bacteria can multiply themselves by means of the binary fission. It means that one bacteria can generate two again different cells. Okay, that is the binary fission. The second one is the budding. It means that a mother bacteria can have a small bud-like structure at the end of the bacterial cell. That is the budding. Then why there is a generation time is generally count whenever we are called the bacterial time or whenever we are counting the bacteria in a dividation. Okay. Now the time required for bacterial cell to divide is called as the doubling time or generation time. It means that from one generation to another generation is time taken for generation is called as the generation time. Okay. Now we can also call it as the interval of time between two cell divisions or the time required for bacterium to give rise to two daughter cells under optimal condition. It is also called as population doubling time. Okay. Now here we can see the example, that is the Escherichia coli. It may take about only 20 minutes to form their next generation. In some cases, that is Mycobacterium tuberculosis, it may take 24 hours to generate their next generation. So consider the productivity potential of Escherichia coli. We can use it in a vast number of manufacturing processes and in our medicinal purpose. Now here you can see how your bacteria can generally use to form the cells. Here at the 0th number of generation there is a 1 number of bacterial cell, and as far as the generations are get increased, you can see that at the 20th generation you can have about 1 lakh of bacteria. Now see, this is the general visual representation of bacterial number. So this is the fastest way of bacterial generation. Okay. Now bacterial growth curve. Now this is the most important thing we have to study in case of bacterial growth, or we can say that the bacterial life cycle. Now the general concept of bacterial growth, when bacterium added into a suitable liquid medium and it incubated, its growth shows some phases, that is called as the bacterial curve. Now consider when you take a single type of bacteria and add into a suitable liquid medium. We have already studied it. Okay. Now that medium, with that medium you have to incubate that bacteria and you can get the growth of that bacteria. Now that bacteria may shows different phase if bacteria count are made in interval after inoculation and plot in relation to time, the growth curve is obtained. Here the log of number of bacteria to the time, which gives us 4 different phases, where the first phase is lag phase, the second one is exponential, or most of the time we are called it as log phase, then stationary and death phase. These are the 4 phases of bacterial life. Where the lag phase. Now the lag phase, where the bacteria do not increase in number, but they are maybe increase in size of the cell. In lag phase, bacteria are new to the environment. Bacteria are new to that overall medium. So that bacteria are trying to adapt to that medium and they are forming a straight line. It means that there is no increase in number of bacteria, but they are just synthesizing macromolecule, which are generally used to multiplication of that bacteria. And the length of bacterial lag phase depend upon the conditions in the original culture and the medium into which they are transferred. Then second one is the log phase. We are also called it as exponential phase. Here you can see the curve is raised by time. The cells start dividing and their numbers increases exponentially. The doubling time is measured during this time. The bacteria are most susceptible to antibiotics during this time. the bacteria stop growing due to increasing nutrients and O2 supply, and they can accumulate of toxic material. Then the next step is stationary phase. Now in case of stationary phase, cell division stops due to depletion of nutrients and accumulation of toxic product. The equilibrium exists between the dying cell and the newly formed cell. So viable cell count remain stationary, where you can get this straight line again. And after that, the last one is phase of decline or death phase, where the population decrease due to death of cells and autolytic enzyme is generally used to secrete by the bacteria. So here you can generally use to get 4 phases of your bacterial growth. First one is lag, where bacteria are trying to adopt that phase. Log phase, bacteria are more active in this phase and give highest population of them. Then stationary phase, where decline phase and the growth phase goes parallelly. So accumulation of toxic material. So the stationary phase is there, and the last one is decline phase or the death phase. Okay. So after that, the maximum cell size toward the end of lag phase is generally observed during the bacterial growth curve. In the log phase, the smaller cells and strain uniformity is observed. In case of stationary phase, irregular staining, sporulation, and the production of exotoxins and antibiotics are done in case of lag phase. Now, what exactly the factors affecting to the bacterial growth? Now, they are temperature, atmosphere, in that case oxygen and carbon dioxide, hydrogen ion concentration, moisture and drying, osmotic effect, radiation, mechanical ionic stress. So these are some things generally used to observe that they are do some severe effect on the growth of bacteria. So this is it for today's session. So for counting of bacteria, how to count the bacteria, how much bacteria can grow in a media, that things we can see in a next lecture. Up till